Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other. So here we will discuss about the regulation of insulin and glucagon production. So the glucagon and insulin antagonistically work, and how they work we will discuss about the endocrinology of insulin and glucagon with respect to the production from the beta cell and alpha cell in the pancreas. So here glucose and insulin antagonistically work. So the glucagon increasing the catabolism, while insulin stop the catabolism and increase anabolism. It means catabolism the breakdown. So anabolism mean the making synthesis of something, which that is anabolism. For example, glycogen formation, the glycogen synthesis from the sugar. So that is known as the anabolism. That is possible due to the insulin production from the pancreas. So here I am drawing the person which that is contain the pancreas with beside the duodenum. So that is this is the pancreas. We can draw with the stomach, and the pancreas showing in the figure is look like this. So this pancreas, the structure is the red color show is the figure, which contain a tail and head and the mid region. So these pancreas work as a exocrinally to produce an lipase, chymotrypsin and trypsin, while and pancreatic amylase. While on the other hand, the endocrine system, which that is the produce hormone into the bloodstream, which that contain a tissue-like structure are present, is known as the islet of Langerhans. And that islet of Langerhans contain a alpha cell, beta cell, and delta cell. So just we will discuss about this is the islet of Langerhans, which contain a different type of cells, and these cells is basically we will discuss one by one. So this islet of Langerhans is looking like this, and that's the endocrine part of the pancreatic cell. So this is used for the regulation and the change the environment. For example, the increase of the blood sugar to decrease by the insulin, while the decrease of the blood sugar to increase by the glucagon. So that is the antagonistically work to each other, this hormone, to maintain and as a homeostasis in our body. So this is the blood vessel, artery and vein, which they are showing the blue color and red color, which supply the blood as well as absorb the hormone. So here the alpha cell, which that is the produce glucagon. So glucagon producing cells increase blood, basically blood sugar, increase blood sugar level into the blood. So that's why it's depend to the decrease of the blood glucose level. So when decrease the blood glucose, that will be increased by the alpha cell produce glucagon, which that is reverse to the glycogen, that is known as the glycogenolysis. So here the insulin production from the beta cell, of the pancreas allow to sugar move in the cell. So the cell will move, allow to absorb the sugar in the presence of insulin, while the delta cell produce the somatostatin, which gives the promotion of growth and development. So that is just like a pituitary gland, which that is produce the somatostatin, is also from the adenohypophysis. So just like that is equal. So here is the blood vessel, which that is we will understand the regulation of insulin and glucagon. So when we will not eat the food, so that during the fasting state, that will be glucagon will be increased, Why glucagon will be increased? Because liver and muscle will act to uh, produce the glucose from the glycogenolysis. So here is the pancreas. When we are not eating food, we are going to fasting stage, and during the starvation stage, here is the insulin level will be decreased, while insulin glucagon level will be increased. So insulin level will be in decrease. Why? Because the insulin is the anabolic hormone which they are used for the anabolism and absorb the sugar into the muscle and liver to synthesis of glycogen. So here is the green color shows that the glucagon will attach with the hepatocyte and myocyte, and after the attachment, which that is primary messenger disease, and to give the signal of secondary messenger, to make a secondary messenger by the cascade of different events to stimulate the production of glucose from the glycogen. So on the other hand, here is one more important. Sympathetic nervous system we will discuss if we will talk about the sympathetic nervous system, which that is also enhance the glucagon. How? Let's here we will discuss about the fight and flight response due to the sympathetic nervous system, which gives the more alert, more breathing, more heart rate, pupil dilation, as well as vasodilation and vasoconstriction in the different region, while the more active and energized person during the sympathetic nervous system. So here is the muscle, and that is the adrenal gland as well as the pancreas are present in the red color show. So here is the adrenal gland, which that is a neurosecretory cell stimulate to produce cortisol hormone. This cortisol hormone will be stimulated by the binding of the pancreatic cell to produce more glucagon, and this more glucagon production is stimulate the more uptake of sugar from the binding with the liver and muscle cell. So that's myocyte and hepatocyte will be allowed to move out the sugar from their cell. So that's glucose level will be high. So that's important. Know that a glycogenolysis will be occur, and that is insulin is important, and that's insulin will be inhibited due to the anabolism inhibition. So this anabolism will be inhibited by the production of glucagon. So the here is the adipose tissue, which that is used for the lipolysis. So the adipose tissue will be decrease the fat, and this fatty acid will be moved into the bloodstream. So basically lipolysis will be occur, and that means the glycogenesis will be formed from the fat into production of ketone as well as the sugar for maintaining the sugar level. So important here is the also sympathetic nervous system, and during exercise that is same mechanism to increase the glucagon. So the glucagon will be increased by the sympathetic nervous system is enhanced during the exercise, for example running. So these exercise will be increase the sympathetic nervous system, mean anabolism will be decreased while the catabolism will be increased. So here is we will discuss about during rest when we will eat and digest this food. So here is the basically when we are eating the food, food will be basically enter into the bloodstream in the form of sugar, amino acid and fat, and this sugar will be allowed to stimulate the production of insulin from the pancreas, the production of more insulin. So the insulin will be produced more. When insulin will be produced, this insulin go into the bloodstream, and when go into the bloodstream, which that is the insulin receptor are present in the adipose tissue, muscle tissue, muscle cell, and liver is also contained, and that means this sugar level is increased during the fat state, and when we will eat the food, so that is here is the adipose tissue, which that is also contain a receptor and also contain a liver and muscle cell. So this receptor will be bind the insulin. So insulin is the primary ligand, is known as the primary messenger, or the facilitate the sugar move inside. So here is the movement of sugar into the tissue by a binding of the insulin receptor into the myocyte or liver cell. So this tissue is here, and this tissue will be bind and the with the insulin to facilitate the glucose molecule to inside for use the Krebs cycle and glycolysis and electron transport chain to produce ATP, either go into the basically adipose tissue to storage. So that is converted into the fat. So that is known as the lipogenesis, the formation of lipids. So here is the digestive system. So we will discuss about the signal of the enteroendocrine cell, which that gives the signal to stimulate the production of insulin, while if during the possibility to produce the glucagon, that is also produce the glucagon from the enteroendocrine cell. So first of all, we should understand the enteroendocrine cell. So here is the large intestine, small intestine, and duodenum. So the small intestine here is the transverse, ascending, descending, sigmoid colon. The large intestine containing. So here the small intestine contain a enterocyte. It is also known as the brush border cell, which contain a brush border enzyme also, and used for the absorption of nutrients, while the enteroendocrine cells are also present. The different enteroendocrine cells produce the endocrine hormone, which that travel into the blood and that's go into the basically here we will just talking about the pancreatic production, that is the stimulation to the pancreatic cell. So the enteroendocrine cell will produce the glucagon dependent insulinotropic hormone. So the insulinotropic hormone will be produced. It is also known as the incretin. So let's talk about the we will explain with the detail to understand how it's work and how to inhibit the insulin production during the fasting stage, and how the glucagon inhibit during the fat state. So here is the blood vessel, which that is present near to the that is present near to the pancreatic cells, pancreatic tissue. So when the glucose level will be increased, so it means the glucose level will be increased. So it means the insulin production is the necessary for the absorption of glucose into the cell to normalize as a homeostasis. While here is the GIP is also present. So first of all, we will discuss about the GIP. So this GIP is also produced due to the increase of the amino acids as well as the food which that we will eat that stimulate the enteroendocrine cell to produce GIP. So this GIP here is the cell, alpha cell, beta cell and delta cell, which that is interconnected to each other and inhibited by each other to production of one hormone, and the other will be suppressed. So here is the B cell and alpha cell and delta cell of the pancreatic island of Langerhans. So here we will just understand first of all the alpha cell. So here is the blood vessel which contain a GIP, glucagon dependent insulinotropic hormone. So that is also known as the incretin. So this is the bind with the primary ligand GIP. So this primary ligand GIP is basically the primary ligand used as a primary ligand, and which that is bind with the GIPR. It means the glucagon dependent insulinotropic hormone, that is known as the incretin. And when we bind, the G protein is attached with this receptor, and this G protein attachment G protein contain a 3 monomer, 3 subunit of G protein, alpha, beta and gamma. So The alpha, beta and gamma are present, which that is the alpha subunit contain a GDP, guanosine diphosphate. So this guanosine diphosphate is basically is use the GTP to produce GDP, and that one phosphate group is transferred to the GDP to release the alpha subunit with the binding of the GTP, and the GDP is produced by the ATP to using as to produce GTP, and that formation will be occur. While here is the adenylate cyclase enzyme used by the binding of GTP with the alpha subunit to detachment, and this detachment to produce the using of the ATP, and this ATP release the 2-phosphate group to produce the cyclic AMP, and this cyclic AMP is the second messenger which that stimulate the protein kinase inactive to activate the protein kinase, and these activate the protein kinase is phosphorylase of the protein kinase. So this phosphorylase protein kinase A activated protein kinase is just a stimulator which that's go into the nucleus and bind with the DNA to, if the sugar will be high, so that's inhibit. If the sugar will be not high, so that will be stimulate the production of glucagon. While here we will just understanding that production of sugar is high during the fat stage, so it means it's a different kind of glucagon like hormone will be produced. It's known as the glucagon and glucagon like peptide GLP-1, which that is released by the exocytosis by this production from the DNA stimulation by the pKa phosphorylase protein. So remember that. And that here is the basically granule will be exocytosed, and this exocytose will be ultimately bind with the GLP-1 receptor of the beta cell of the insulin production beta cell. So here let's talk about the insulin producing beta cell. So the insulin producing beta cell is here, which that's contain a GIPR receptor, also same just like a glucagon releasing cell contain it. So this basically GIP will be bind with the receptor to the produce the different the cascade of different events to produce cyclic AMP is the second messenger. So it means sugar is also present, and that sugar is going inside by the GLUT2 channel by the using of one sodium ion move inside also, and that sugar move inside by the binding of the enzymes known as the glucokinase to by the also use the phosphate group to produce the glucose 6 phosphate, and this glucose 6 phosphate is by using by the glycolysis to produce pyruvate. So this is the separate mechanism, while we are understanding the GIPR receptor to bind with the GIP is the different mechanism for stimulation to release the insulin. So here is the mitochondria. When pyruvate move inside to stimulate the TCA cycle, and this TCA cycle tricarboxylic acid cycle to go in the electron transport chain to produce ATP, and this increasing of the ATP here is the potassium ATPase pump or potassium ATPase channel, which that is the potassium move outside is the inhibited by this ATP, because the ATP dependent potassium channel which that is used for the movement of potassium outside. So the membrane depolarization will be occur, and this membrane depolarization will be allowed to stimulate the VGCC is a L-type calcium gated channel, which that's allow the calcium move inside from the outside. So the constriction of the calcium move inside is more, and that means you know about that the basically acetylcholine receptor is the acetylcholine neurotransmitter. It look like this, and that is the basically calcium which that is the exocytosed by the this insulin granule which that is produced from the. So it means here is the insulin granule is stimulated by the calcium ion, while on the cyclic AMP is also stimulate. How it stimulate we will discuss, just understand. So here is the basically metabolic signal which that is stimulated by the production of glucagon and glucagon like peptide which that stimulated by the binding of the GLP-1 receptor. So this GLP-1 receptor is also present in the beta cell. So the GLP-1 receptor is the glucagon-like peptide 1 receptor which that bind and to produce a same cascade of different event to produce a cyclic AMP. So this cyclic AMP will be stimulated when go into the nucleus to produce a messenger RNA, and this messenger RNA will that is production of messenger RNA is known as the transcription, which that is the DNA will be produced RNA into the cell, into the nucleus. So the inside of the nucleus, basically inside of the nucleus, the RNA polymerase enzyme, DNA helicase enzyme, as well as the basically transcription factor are present, which that is bind by the cyclic AMP, the cascade of the activation of the protein kinase A phosphorylase, which that is we had also understand in the previous diagram. So then produce the transcription by the transcription to produce a messenger RNA, and this messenger RNA is produce the insulin by the go into the rough endoplasmic reticulum to Golgi bodies, and that's maturation, and that is known as the translation, while the transcription is also lead to produce UCN3. So the UCN3 is the important to stimulate the delta cell. So here the insulin will go into the bloodstream. When go into the bloodstream, that means the allow to move the sugar, facilitate the sugar into the muscle cell or liver cell. So it means here is the delta cell which contain a CRH2 receptor. So this CRH2 receptor is receive the UCN3, and this UCN3 will be receiving to stimulate the same mechanism of the cyclic AMP production, that is the second messenger. So this second messenger is basically to stimulate the nucleus to produce more somatostatin. So the what is the function of the somatostatin? And this is the exocytosed, and that function is the growth. So important to know that here is the alpha cell of glucagon, glucagon producing cell which that is bind with the somatostatin to inhibit. So inhibition. So the cascade of different events will be occur by the binding to produce a second messenger is known as the cyclic AMP, and that's lead to produce the protein kinase A by the activation to phosphorylase, which that is the same mechanism to produce, and that means that will be go into the nucleus, and that's nucleus will where the transcription factor are present, which that is suppressed by this mechanism, because somatostatin receptor is used for the inhibition of the production of glucagon. So when go into the nucleus, here is the nucleus, this protein is used for the suppress or decrease the production of glucagon by the transcription factor to inhibition. So the glucagon will not increase. So that's why the glucagon is a catabolic hormone, while the somatostatin and insulin is the anabolic hormone. So the somatostatin is used for the production and growth of the cell or growth of the muscle, etc. So that's why the suppression of the basically or inhibition of the glucagon production due to the somatostatin while the somatostatin is produced by the stimulation of the beta cell of the insulin production. So the insulin stimulate while the glucagon will inhibit the somatostatin production. So the same mechanism, because when glucagon will be increased to inhibit the insulin production by this activity, while because glucagon will bind with the glucagon receptor with a beta cell of insulin to inhibit. So they same mechanism which that is occur, just like stimulation, and that is the basically inhibition is the same like. So this is the insulin and somatostatin which that is inhibit the glucagon, while the glucagon is also inhibit the insulin and somatostatin production. So the same antagonistically work. So this is the basically insulin and somatostatin is basically another way. So here is important thing is that is basically a GLP-1 which that is produced, and that's go in the binding with a beta cell, and that's beta cell will produce the insulin as well as UCN3, and which that is bind insulin is used for the muscle growth and lipogenesis, as well as the production of basically attached with the liver to facilitate the sugar move inside, while the UCN3 is also stimulate the somatostatin by the delta cell of the pancreas. So here is a basically cascade of different events will be occur by the first ligand and the second messenger, and that's go into the stimulate. Here is the nucleus to produce UCN3 and insulin by the translation of the producing. Here is the translation, and that is the messenger RNA produced is known as the transcription, and this translation is used for the production of the insulin, and this insulin will be packaged up by the endoplasmic reticulum and Golgi bodies to maturation, and this is phase to transfer of the Golgi bodies to maturation, and that is allowed to exocytosis by the stimulation of the calcium ion, which that we had also understood about that things. So that's stimulation. So important thing is that, while here is the basically that is basically here is the exocytosis to go into the bloodstream, and where the need for the insulin for using as a energy when go, or as a storage in the form of fat in the form of adipose tissue, that insulin will be bind with the insulin receptor to allow to facilitate the movement of sugar into the cell, into the cell of the tissue. So here the GIPR receptor is also present in the glucagon producing cell and insulin producing cell. So this is the mechanism, both is contained, but GIP receptor are bind with the alpha cell to inhibit the production of glucagon, while basically the production of the while the other glucagon and glucagon like peptide GLP-1 produce, and this produce to stimulate the insulin producing hormone. Insulin producing cell is the beta cell. So here the glucose is also enter inside, so that means the sodium ions will be used for it, while here is the potassium will be inhibited, and the inside is the constriction more to membrane depolarization, and that's allowed to calcium ion move inside to balance it, while the increase the calcium ion inside, so the L type calcium catalytic channel will be open. So that's exocytosis of the insulin by the stimulation of calcium ion, while the increase of the insulin due to the translation of the protein of insulin will be increased. So that is also stimulate, because the increasing of the insulin granule. So here is the ATP is increase is the totally depend to the increase of the sugar level in the blood stream. So next video we will also understand about the fat impact on the beta cell and alpha cell and how it's work. On the other hand, the amino acid is also used for it, so we will also understand about that. But here you can understand just basically the second messenger concept and the alpha cell, beta cell and delta cell. So in summary, we will understand just in summary. So the alpha cell produce the GLP-1, which that is stimulate the beta cell to produce insulin in the presence of high level of glucose during the fasting fat state. While the fasting state, it is the opposite. On the other hand, the beta cell is also stimulate the delta cell of the pancreas to produce the somatostatin, and the somatostatin is the important inhibitor of the glucagon production and the GLP-1 receptor. So when glucagon GLP-1 will be not produced, it means the insulin also not produced. So that means when insulin will be produced to stimulate UCN3 binding to produce, and that's ultimately insulin inhibited and the somatostatin will be also produced.